Hello everyone, welcome back to one more video session. In this session, I'm going to cover three small concepts. All these concepts are important for 1 mark as well as 2 marks. For example, the first concept, the characteristics of a good genetic material or the properties of a good genetic material. You could get this question for 2 marks. You have to write only 4 points. These are all the points you have to write. Very simple, very easy. Properties. If you take DNA or RNA, we know that DNA is the genetic material. We know that the DNA also got its own properties, whereas the RNA also got its own properties. But DNA is far more better than RNA in its properties. That is what we know about it. But here, to understand this concept, we have to assume that we don't know anything about the genetic material. We are just assuming, okay, there is one genetic material and it has got certain characters. And ultimately, because of these characters, we can call them as or call it as a good genetic material. Like that, you have to assume. That's it. The first property of a good genetic material or ideal genetic material is it should be able to replicate itself. What is the meaning of replication? Replication means production of its own copies. Why it has to produce more copies? You know that during cell division, the chromosome, which is nothing but DNA should be shared equally among the daughter cells. That's what it has to replicate. It is making sure that its sequences or its information is being shared in each daughter cells, so that the daughter cells will carry the information for the production of the specific type of proteins. In one daughter cell, one type of specific protein is produced. The same specific or type of protein should be produced in another daughter cell because it is given by the same mother cell. That's it. Okay, so that is actually the thing able to replicate itself. And in our previous video session, we have discussed about the structure of DNA and RNA, and I said DNA is a better genetic material compared to RNA because RNA is highly unstable whereas DNA is stable. You know the reason behind it. That's what, if anything should be called as a genetic material, any chemical as a genetic material, it should be structurally and chemically stable. Structurally stable, yes, it should withstand temperature, high temperature. At the same time, it should not react with any other chemical. If it reacts, obviously the structure or the chemical nature will be lost. It cannot be a good genetic material because the information is stored in a genetic material which could be lost. That's it. It cannot pass its information to its like offspring or its generation. That's it. And then undergo slow mutation. Yes, you know about the mutation. There are so many mutations, like fast mutation is also there, slow mutation is also there. Slow mutation is nothing but V. Because of slow mutation, you could see diversity in living organisms. Because of that, you could see a variety of living organisms in this earth. That's it. This is the thing. So it should undergo slow mutation. Slow mutation, that means a new sequence will be added gradually or some sequences will be deleted gradually. It is not a fast process. It will not take place within one generation. It takes millions of generations. Got it? So this is the thing. For this process, it has to take lot of time. Slowly, it has to undergo mutation and ultimately a new species will be produced. This is the thing. Next, it should express Mendelian characters. You know about the Mendelian characters. That means, you know, the transfer itself to the next generation. That's it. Like the DNA should, or DNA or RNA. I'm not talking about DNA here. I'm talking about a good genetic material. The genetic material should be able to pass its information to the next generation. That's what. Transfer itself to the next generation. That's it. For example, you take a single celled organism, let us imagine like a protozoa like amoeba. Amoeba contains the DNA and it undergoes cell division to produce 2 daughter amoebae. In each amoeba, that is daughter amoeba, you could see the DNA which is given by the mother, correct, the parent. If you look into the sequence present in the DNA of the one daughter amoeba and another, we compare it with another daughter amoeba, both are same, exactly same. That means the DNA has shared or like transferred its information into the 2 daughter amoeba. That's it. So that is transfer itself into next generation. This is the thing. So all these are very, very important 4 properties. If any chemical exhibit all this property, then we can call it as a good genetic material. That's it. So DNA fulfills all these properties, that's it, or exhibit all these characters. So DNA is a very good genetic material compared to RNA. Now let us learn about the differences between DNA and RNA. why DNA is preferred as genetic material than RNA, or why RNA is not preferred as genetic material. What are the drawbacks of RNA? What are the advantages of being DNA? So let us see that. Simple, it's very easy. We know about it. That is, when we were studying about the structure of the pentose sugar of RNA as well as DNA, you know that in the second carbon atom or in the second position, you could see that in RNA there is oxygen, there is hydroxyl group, which is highly reactive, makes the RNA unstable and makes the RNA degradable too. That's it. And whereas in DNA, that in the second position of its pentose sugar, the O is absent, the oxygen is not there. That's what makes it deoxyribose sugar. That's it. Which makes the DNA stable. RNA has free hydroxylic group at the second carbon atom in its pentose sugar and makes it labile, that is weaker or unstable, and the RNA could be easily degradable. This is the important difference between DNA and RNA, whereas DNA is better structurally as well as functionally and even chemically compared to the RNA because of this one important property. Remember it, it's very important for your NEET. Remember it. Then RNA, as I said, like it is unstable, it is not chemically stronger, and it mutates faster. It mutates faster. There is a reason behind it why RNA mutates faster, because you look into the fourth sentence here. They do not have the thymine. In the DNA, adenine, guanine, cytosine, thymine is there. Thymine provides more stability to DNA. Whereas thymine is absent in RNA. Instead of thymine, uracil is there. Uracil now replaces thymine because of the uracil, which makes RNA bit weaker compared to DNA. That's it. So RNA mutates faster. Remember it. Okay, so we have learned two important statements. Then yeah, some RNA, they have the catalytic activity. If you look into the particularly focus into the nucleus, you see so many RNAs, you could see a division of labor, a good division of labor. Some RNAs are meant for carrying information from DNA to the cytoplasm. Some are required for catalyzing a particular activity. What is the meaning of catalysis? Catalysis means it is a type of a factor which now reduces the amount of energy in a particular reaction, or helps the reaction to go faster. Correct? Okay, so this is the thing. Catalysis is nothing but enzymes. Sometimes the RNA behaves as if they are enzymes. So what you call them if RNA behaves as enzymes? Please remember it. It's very important. I'll write here. They are called as ribozymes. Ribozymes are nothing but RNA which acts like they are enzymes. They are not enzymes. They behave as if they are enzymes and they help in catalytic activity. If RNA helps in catalytic activity, then obviously it is highly reactive. It cannot be a good genetic material. In the sense boss should be boss itself. Correct? So boss cannot be an employee. The same thing is applicable here. The RNA behaves as if boss as well as employee. So it cannot be a good genetic material. And it's weaker compared to the DNA. DNA stays there itself. It is stationary. It will not do anything else. It just stores information. That's it. It's very specific. But in the case of RNA, it is not specific. It is showing division of labor, which is meant for different types of function. So this is the thing. Whereas the absence of thymine in RNA makes it unstable. That's what we have discussed. Yes, because of that RNA mutates faster. RNA is shorter. Yes, if you look into the length of the DNA and RNA, the DNA is very long, RNA is very short. RNA normally acts like a messenger, it just carries the information which is stored in the DNA from inside the nucleus to outside the nucleus, that is the cytoplasm. That's it. It is just acts like a messenger from DNA to the ribosome to synthesize a specific protein. Because of its shorter nature, it cannot be a good genetic material. That's it. So it can be used for passing the message from DNA to the ribosome or DNA to synthesize a particular protein. That's it. So it can sneak in between the nucleus and it can enter into the cytoplasm. That's it. So this is all about why DNA is preferred than RNA as genetic material. That's it. So that's all about it. Now let us learn some basic information about DNA. the last small concept, that is you have to know the total number of base pairs present in the DNA. It could be single stranded DNA or double stranded DNA of an organism. So there are 4 examples which are given in your NCERT textbook. So you need to learn this. It's very simple. You just have to remember this chart. I have written the type of the organism here, and it is the type of the DNA. You could see here I have mentioned circular SS. SS means single stranded DNA. The DNA could be single stranded in only viruses. When it comes to higher animals or organisms, obviously the DNA should be double stranded. You have to remember this. Here I have written linear. Linear means straight, whereas circular, linear. This is the type of the DNA, and this is the strands. How many strands are there in a DNA? In this case it is single strand, in this case it is double strand. Okay, double strand, double strand. Whereas here I have written the total number of base pairs. Please, very simple, it's very easy. The base pairs means, for example, if I write here, let us assume that this is the double stranded DNA, and these are all the nitrogenous bases. Correct. Okay, so these 2 strands are now joined by the hydrogen bonds, and you could see T here, 1 T, and this is another T of another strand. They are linked by hydrogen bonds, and here you could see adenine, and here you could see thymine. They are complementary to each other, or it could be sometimes cytosine, guanine in the case of DNA. That's it. Okay, so if you count it, see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Total number of base pairs in this DNA is 11. That's it. So if you look into the circular single stranded DNA, if you count the total number of sequences or base pairs, that is what I have written here. The total number of base pairs in this particular DNA present in this organism. Bacteriophage 5x174 contains or has a circular single stranded DNA. If you count the total number of base pairs, circular single stranded DNA. See, single stranded circular. This is actually the DNA of the bacteriophage 5x174. If you count it, there are about 5,386 base pairs. That means it is the smallest DNA, correct? Smaller DNA, which is the base pairs are very less. 5,000, very less. Let us go to the next one. The bacteriophage lambda, another virus which infects the bacteria, which is called as bacteriophage lambda. Okay, so which has got a linear double stranded DNA. So linear, see, linear, straight line, double stranded, you could say 2 strands. And if you count the total number of base pairs, it has got 48,502 base pairs. So a bit larger compared to this, correct? Okay, so it is like longer DNA which has got approximately 50,000 base pairs, but you have to learn this number 48,502. Just mug up. Okay. The next one is Escherichia coli. It's very common bacteria. In second year PUC, we are going to study about it in detail about Escherichia coli in microbes in human welfare chapter. You could see circular double stranded DNA. See, circular double stranded. See, 2 strands are there. And if you count the total number of sequence, it is more than 46 lakhs. 4.6 into 10 to the power 6 base pairs. Almost 46 lakhs. Okay, remember it's very long. Okay, it has got almost like 46 lakhs of base pairs. The next one is humans. Here I have selected human gamete. It could be ovum or it could be sperm, which is a haploid cell that is having only 23 chromosomes. If you take all the chromosomes, obviously chromosomes are now separated, that is 23 units, but if we just join them, obviously it is a long thread, correct? Okay, that is linear double stranded DNA, linear double stranded. If you count the total number of base pairs, it will be more than billion, that is 3.3 into 10 to the power 9 base pair. Please remember it, it's very easy, but you have to know the total number of base pairs and the type of the DNA present in these particular organisms. That's all about it. So we have learned 3 small concepts. All are important, could be asked for 1 mark or 2 marks. Learn it.